I'd like to welcome Denise O'Mara from the Waterford Institute of Technology. And Denise is a lecturer at the Waterford Institute in the southeast of Ireland. And Denise has studied red squirrel genetics for her PhD and has since applied genetic tools to help study a range of species, including squirrels, pine marten, otters, badgers and bats. So over to you, Denise.
Hi, thanks very much for inviting me to speak to you today. It's lovely to be here. The work I'm going to talk to you about is a collaboration between ourselves in Waterford Institute of Technology, the University of Salford, and the Red Squirrels United group. So the work has been funded by Red Squirrels United, and what we're actually doing is looking at some of the grey squirrels that have been culled around Anglesey and North Wales, and to see if we can understand a little bit about their genetics and to see how we might be able to use it to help inform management and, I suppose, help understand how they've actually moved back into the island. So the thing is, we can use genetic tools to help us understand a little bit about the conservation of a native species, but we can also use it to understand how we might actually manage an invasive one. And previous studies have shown that, like in a lot of situations, a one-size-fit doesn't suit them all. And each species has to actually be studied and understood individually in order to be able to use the tools to make a bit of sense out of them. So basically, each species needs a tailored approach, and that's what we're trying to do in this project. We're trying to see what different genetic methods we can actually use and what each of them can actually tell us and how we can use it to inform future conservation and management. So this is just a little map of the area that we're particularly interested in. So we have Anglesey mapped out there in red, we've Gwynedd and we've Clough-Éineog mapped out as well. So we have actually about 400 samples in total from the area. So all of these samples that have been culled by the conservation groups in those areas, and we've extracted the DNA from them, and we're using different tools to be able to see which ones are actually telling us the most information. So a little bit of a background information about squirrels in Anglesey. They were first documented on the island in 1966. An earlier study showed in 1959 that there weren't actually any grey squirrels there at the time. And in 1998, according to Craig's work, there was actually between 3,000 and 4,000 grey squirrels on the island. And through massive control and eradication efforts, they managed to completely eradicate grey squirrels from the island. So I can only imagine how much work was actually involved in that. But actually over the years, grey squirrels have continued to come back onto the island. And as we've heard already, they've likely re-entered from the mainland, either through the two land bridges, or they've possibly swam, or we may actually have had people intentionally introduce them there. We're not entirely sure. And a study by Lisa Signorile and Craig back in 2016 used nuclear DNA, which is one type of a DNA marker, and it showed that some of the genetic similarity, that there was actually similarity between grey squirrels in Anglesey and on mainland Wales, but also there were some genetic differences, particularly in the southwest of the island there that I have marked. And the actual origins of the population were left a little bit unclear. So I think the important thing to mention here, before we get into some of the DNA stuff, is that genetics by itself won't tell you a whole lot. You really need to use both the ecology and the history of the species to get a good understanding of what it can actually tell you. So it's only one, I suppose, tool in the box that can help understand something. And in isolation, without understanding the history or the ecology, it's not going to tell you anything. So if we have a think back to some of the history of grey squirrels in Wales, for instance, the first records of grey squirrels in Wales went back to 1828. And we actually think that population died out at the time. and it was 70 years later before another population was found around Wrexham. And then we know also that grey squirrels were introduced into northern England around 1876 into Hinbury Park. And one of the biggest, and I suppose one of the most influential introductions was created, I suppose, by a guy called G.S. Page of New Jersey. And he was the person that introduced the grey squirrels into the Woburn population. And that population was used to basically restock most of Britain with, or well, introduce grey squirrels into most of Britain. So I actually have just a table there of from Monica Shorten's 1954 book. And it basically just lists all the historical introductions that we know of grey squirrels into Britain. We have a number of individual introductions from the US. There's 1, 2, 3, 4, 5, 6, about 7 or 8 individual introductions from the USA. The Woburn population appears significantly, so that was used over and over to restock the British population, or to stock it originally. And then there was other populations from London and the zoo as well that were used to basically move squirrels around around Britain. So it wasn't just one introduction. We were dealing with a lot of introductions that took place, and a lot of what we would see would have caused a lot of genetic mixture as well going on there. I suppose it's important to think as well about what was going on with the red squirrel at that time. And we often assume that the red squirrel declined as a consequence of the introduction of the grey squirrel. But actually the red squirrel was in decline long before the grey squirrel was introduced. But the impact of the grey squirrel had a significant negative impact. But it's important to remember that their population was declining due to habitat removal. So you would have had oak that would have been used to build boats for the Navy, wood that was used for charcoal to fuel the iron, lead and glass industries. And then we had a lot of environmental instability around the time of the late 1700s and 1800s as well, where there were severe winters, and it's quite possible that the squirrels were impacted by that as well. Also, we were doing a lot of moving and selling of red squirrels around that time. So the Victorians seem to love selling and moving red squirrels around. And in Monica Shorten's book, she writes that about 20,000 red squirrels were sold annually in markets in London in the 1800s. And these red squirrels originated both from Britain and also from continental Europe. And it's likely that we've probably imported some of the diseases and things like that that might have actually had an impact on the population today as well, because we were moving squirrels around so much at the time. So different genetic markers can help reveal different aspects of the situation. So the choice of genetic marker can tell us quite a bit, and I just have 2 examples up here. The first one is our mitochondrial DNA haplotype, and basically this is a part of the DNA that will tell us a little bit about the strain or the genetic type of the animal. It evolves very slowly, so it actually retains a lot of the genetic history or the genetic footprint of where the animal had come from originally. And then the other marker that we have is called nuclear DNA, and it's composed of these things called microsatellite markers, which we've used. And basically these microsatellite markers, we'd need a panel of them to look at a population. And what we do is we look at differences in the number of alleles across individuals. And we build up a genetic fingerprint for the different animals, which allows us to tell the difference between one animal and the other. But it also tells us a little bit more about the contemporary genetic diversity of the animal, telling us a bit about how diverse they are and even how related they are to other animals. So with that in mind, it's important to build on what we already know. So a previous study that was conducted in the University of Cumbria by Claire Stevenson Holt and co-authors, they looked at grey squirrel mitochondrial DNA in the Cumbria area, but also compared the DNA from there with other samples from the south of England in Alice Holt, but also some populations in northern England and up into Scotland as well. And what they found were 12 different mitochondrial DNA haplotypes within the population. And if you have a look here, this thing is called a network diagram. And this network diagram basically connects up the different haplotypes by how similar they are to one another. So the closer one haplotype is to another, the more genetically similar it is to it, the more likely they originated from the same source population to begin with. And what they found was that these haplotypes, even though you might have had some of them down in the south of England, they could often be genetically similar to haplotypes found elsewhere in northern England and Scotland as well. And then in terms of microsatellites and what we know about microsatellite DNA from previous studies, a study from Lisa Signorile looked at the nuclear genetic diversity across all of these different populations in the south of England, into Wales and a little bit into northern England, and also a population from Ireland as well. And what she did then was she did a thing called a population assignment. And that's this little chart up here. And you can do this in a program that basically assigns every individual in your data set to its most likely source population. And generally, when we use this in wildlife samples, we find very few populations. But in the grey squirrels, what we're finding are lots of distinct populations, which suggests that grey squirrels didn't actually move around a whole lot once they'd actually been introduced. So we can see here, if we look at the North Wales and North England population, there's some genetic similarity there. And again, if we look into Cornwall, there's a pink and a blue cluster here, and the pink and the blue cluster also continue up into the Midlands. But generally, what we would say is that having been introduced to various spots by people, grey squirrels didn't actually intermix a whole lot. So the problem is both of those studies use different samples and one genetic marker each, so we can't really directly compare one with the other. They both tell us some interesting stuff, but neither of them are directly comparable. So that's what we were trying to do with this study, was to see if we could actually genetically compare one with the other. So what we're working with are about 400 grey squirrel samples from Anglesey and North Wales. We have a mixture of tissue and hair samples from the various culls, a couple of incidentals and things like that. And we're using both these mitochondrial and nuclear or microsatellite markers to help us get a better understanding of the population. So in terms of the mitochondrial DNA, what we found so far are 6 different strains or haplotypes within the North Wales population. And interestingly, what we can see here is that the dominant haplotype in Anglesey, this orange one, that's found right across the border there in Gwynedd. And it's not, however, found in Clocaenog, but this other haplotype, this dark grey haplotype, is also found within North Wales, both in Gwynedd and Clocaenog. So that would suggest to us that grey squirrels, just with this type of analysis, are simply moving from Gwynedd back into Anglesey. Now, with this type of DNA, it's not telling us a whole lot about the contemporary genetic diversity, so that's why we're going to use the microsatellite DNA as well. And that might tell us a little bit more in terms of how these populations are genetically structured, like what we had with the Signorile paper. But I think so far what we're saying is that they're just moving simply, and we don't have any long dispersal movements or anything like that taking place. So what we then wanted to do was compare what we found in North Wales with what's been previously found throughout Britain. And what I did was I compared the haplotypes that we found with those that have been mapped previously by Stevenson Holt in Cumbria and Northern England. And what we see is quite a bit of genetic diversity across the UK. So if we concentrate just on the Wales samples initially, we'll see that the haplotypes that we found in Anglesey and in Gwynedd, those orange, kind of peachy coloured ones, they haven't occurred elsewhere in Britain, or at least in terms of the samples that we have available to us. This dark green haplotype, which we see in Gwynedd and in Clocaenog, is also in northern England. And we see that this speckled haplotype, which also occurred in Gwynedd, is all the way up here in northern England as well. So we do see that there is quite a bit of movement, and it probably suggests different introductions that would have taken place over the years. We also have quite a bit of similarity going on with, in terms of this haplotype occurring into northern England and into Scotland. And this pale green or grey haplotype down here in southern England, that comes all the way up to Scotland. And the only way that got there is just by people having moved them around. So again, what I did was we compared our new haplotypes that we found in Wales with those that have been recorded previously by Stevenson Holt, and we built a network diagram. And like I was saying, this network diagram connects our haplotypes by how genetically similar they are to one another. And all of this kind of, I suppose, these little black nodes in here, these ones happen to be what we call missing haplotypes. So the software basically computes gaps in it. And that makes sense because we would have had introductions from various parts of America, but not necessarily the entire population. So what we're seeing here are gaps in distribution in terms of the genetic diversity that should be available. And if you look down here, these ones stick out a little bit as well, which suggests that they probably came from different areas. So this is just another view of the crossover of haplotypes there. So I decided I would look at some of the work that's been going on in America as well, to see if we can actually have a look in terms of where the British grey squirrels sit in terms of the American picture. And there was a study done by Nancy Moncrief et al. In 2012, and they published DNA sequences or DNA mitochondrial haplotypes of grey squirrels from across their distribution in the eastern United States, where they're actually native. So if we look here, all of these red ones, these are all the American haplotypes, and I have some green ones dispersed throughout, and they're the British ones. So interestingly, we've actually got British haplotypes scattered throughout, or well, mostly throughout what we know about the American diversity of grey squirrels throughout the whole eastern United States. And within Britain alone, we have haplotypes that are everywhere in America. So that would suggest that we have a very diverse source of grey squirrels in Britain, which probably adds to the problem of trying to eradicate them. And it also adds to their invasiveness because we have quite a diverse population. Interestingly, we have some haplotypes, this one here and this one here, which are shared. So that means it occurs both in America and it occurs both in Britain. And that means, luckily enough for us, that the Moncrief study actually published some of the same haplotypes and probably sampled from the same areas where we had some of our animals actually introduced from. So I went ahead and I decided I'd look at that in a little bit more detail. And what I've done here is I've just color-coded the haplotypes by state. And I hope you can read this little legend down the side. But basically what I've done is I've color-coded the British ones red, and then I have Virginia in green, and I have Maryland in kind of a yellowish colour. And they're the two I want you to kind of concentrate on at the moment, because if you look up here, this is a red one, which is British, and this yellow one, which is from Maryland. And if you remember, when I was talking about one of the biggest and most successful sources of grey squirrel introductions into Britain, we said it had come from New Jersey. And Maryland is the next state down from New Jersey. So what we have now is evidence of the population where it had come from in the States. So we have you can see a lot of the Maryland haplotypes are around here, but when we come over here, we have another haplotype shared with America and Britain, and that's a haplotype from Virginia, again, the next state down, which again shows that while we've got a lot of diversity within Britain that hasn't even been recorded in America, and it's scattered throughout America, we can also actually source some of the animals where they've actually come from multiple states across multiple areas. And like some of these are from really broad geographical areas as well. If you come out from this one, for example, this is a haplotype from Louisiana, and it's just, there's only a couple of small mutations between them. So again, it kind of reiterates how diverse the British population is, that it can actually sit in with all of these animals from across the United States. So just to give you a quick glimpse of a map here, just to give you an idea of some of the areas we're talking about. Maryland is highlighted up here. Virginia is below it. New Jersey is just above it, from where we know we've definitely had introductions from, but we don't actually have any samples from New Jersey to directly compare them to. But Maryland is a pretty good second place there. But if you have a look here, like we've actually got, samples that are looking like they're not that genetically distinct from animals all the way down Louisiana. So that's a massive geographic stretch of space. And I think with that in mind, it might help explain some of the difficulties that we're experiencing in terms of grey squirrel control. So what we can conclude from that is that the British grey squirrels share ancestry with US squirrels across multiple states, not just from individual areas. We still have a lot of work to do. The data suggests that grey squirrels are simply recolonising from Anglesey from the nearby areas. Some of the haplotypes we found in Wales also occur into North England and Scotland, but 3 of those haplotypes were unique to Wales, North Wales and to Anglesey. I would think that if we had samples from maybe Mid Wales or South Wales, that we'd probably find some of those other haplotypes in that area as well. So I think it might be worth kind of expanding out a little bit to see if we can get a better grip as to how grey squirrels are dispersing across Wales in general. What we see is that the British grey squirrels are very diverse, with haplotypes shared with animals from Maryland and Virginia, and some not documented in the US at all. So obviously we have animals that have come from maybe the same state, but also other states as well that just haven't been sampled in the US. So we probably have a lot of information that's relevant to the grey squirrel within its native range as well. So we need to finish completing our microsatellite analysis. We've just started that. We've got a full panel fully developed, and we're rolling that out at the moment. And it'll be interesting to see how our microsatellite data is going to compare to the mitochondrial data, and if it's actually going to add anything else to the picture, whether we'll actually get a better idea as to pinpoint individual locations as to where animals are coming from. The microsatellite data tends to give you a little bit more of a kind of a robust insight, so kind of a greater insight, whereas the mitochondrial data will give you kind of a bigger overview while retaining a lot of the kind of the history and stuff like that as well. And then we want to compare the results that we get with the microsatellite data with what was previously published in Signorils and Shuttleworth's data in 2015 to compare levels of genetic diversity to see if perhaps the culling has impacted the overall genetic diversity in the region, and if that might have an impact on the movement of the animals as well. So that's it. Thank you very much.